Sure. So I'll give a super brief introduction to Marc. So Marc, his background is more in kinase signaling and the biology of kinases, and joined our lab just over 2 years ago, and has been adding to that by learning a lot about the more computational side, and has been leading this project in the lab. That's a big effort with a lab at MIT, Forrest White, and then a clinician, Eric Haura, to look at a long-standing interest of ours, this signaling from a receptor AXL in lung cancer drug resistance. So with that, yeah, Marc, you can take it away.
Okay, thank you very much. Actually, I think it's the first time that someone introduced this to me, and it was very nice. I liked it. And okay, so yes, as Stern said, I'm a third-year PhD student in the Meyer lab, and today we'll be talking about our efforts into finding out what are the AXL downstream signaling pathways or components that lead to EGFR-targeted therapies in lung cancer. So as a brief overview, what is cell signaling in this, and specifically in this context in which we study phosphorylation-based communication? So cells are able, one of the ways in which cells are able to respond to stimuli in their extracellular matrix is by using cell signaling cascades through phosphorylation. So two receptor tyrosine kinase monomers dimerize when their ligand is present in their surroundings, and then upon dimerization, they transactivate each other, the two monomers, leading to an active receptor, which will start a downstream signaling cascade by recruiting a bunch of kinases and other signaling molecules, which eventually will lead to a specific phenotype. However, this gets very, very messy when you start thinking about the number of possible receptors and possible downstream molecules. And what we are interested here is in using systems approaches to really find out, given this input or this ligand here, what are the outputs that this ligand triggers, and what are the key 1, 2, or 3 signaling molecules enabling this connection. However, it's also very important to mention that it's not only the amount of receptors that cells have, but also the same receptor can lead to different phenotypes through different signaling pathways, which can even complicate things more. But again, using systems biology should help us figure this out. Another aspect to cell signaling is the how kinase-substrate interactions occur through many things. Many factors such as colocalization. However, one of the the factor that we're incorporating in this project is kinase specificity. For example, here, if we take SRC, which is a non-receptor tyrosine kinase, this is the structure of SRC, and here we have a cartoon of the substrate, which is here shown. So the docking site of the substrate is shown here in yellow. This interaction is enabled by the immediate surrounding amino acids of the phosphorylation site. And by running biochemical assays, we can find, okay, this kinase prefers to have isoleucine at -1. So if 0 is the position that, or the residue that is phosphorylated, the tyrosine that is phosphorylated, SRC prefers to have isoleucine at -1, phenylalanine at +3, and a whole bunch of acidic amino acids in the C terminal of the motif. And this is very important to keep in mind for my next slides. Okay, but stepping back, what we're interested here is to study bypass resistance, or in general drug resistance in lung cancer, and more specifically bypass resistance. So many lung cancer carcinomas are driven by the receptor EGFR. However, when we block EGFR, this leads to, even though it leads to initial effectiveness in clinical settings, there are many means of drug resistance. And bypass resistance is the activation of other receptor tyrosine kinases that are able to activate the downstream signaling components that were activated by EGFR before, and additionally, maybe a further signaling components driving to growth and possibly further malignant phenotypes such as EMT, metastases, or immunosuppression. And we're very interested in one of these receptors that enable this bypass resistance mechanism called AXL. AXL is a very special or atypical receptor tyrosine kinase in the sense that it requires a very unique set of conditions to become fully activated, which makes it hard for us to study in vitro settings. Previous work in the lab showed that for AXL to become activated needs to have localized Cas6, as you can see, which leads to clustering of the receptor in very special in very defined places of the plasma membrane to become active, as we can see here. And to add another layer of complexity into this mechanism, AXL has also can bind to Gas6, but also to PROS1, but Gas6 is much higher affinity. And on top of that, apoptotic cells, usually apoptotic cells display phosphatidylserine, which is a lipid on their cell membrane, which can bind to Gas6 to even activate the receptor even further. Okay, so to double a bit on the biology of AXL or the TAM receptors. So AXL, MERTK, and TYR3 are the TAM receptors of the same family that share these characteristics that I've been talking about in the previous slide. And they're defined as hemostatic regulators in adult tissues because they take care of, since they bind to apoptotic cells, they are expressed in macrophages and other phagocytic cells and take care of their elimination. So I think I have to speed up a little bit, but yeah, their expression is very broad. They're present in the nervous system, in the reproductive, vascular, hemostatic, and hematopoietic, and immune systems. And very importantly, they have been established to be important in resistance and metastasis. And a few of their signaling components have been established, but in very different contexts and can get a bit complicated. And also, there's more and more interest in their effect on immunosuppression. However, what their effect is in immunosuppression, whether cancer cells take care of these if cancer cells use the TAM receptors to suppress the immune system in cancer is not well established yet. So again, what we're interested here is in finding out and using systems approaches to define what are the key signaling pathways or molecules enabling cancer growth, cell cancer growth. And in lung cancer cells or in EGFR-dependent cell cancer cells, it was established that, for example, ERK and AKT were the master regulators of resistance. And previous work in the lab also established that this can be partially true depending on the settings, and they used a whole bunch of computational methods to find out that junk actually was explaining part of the phenotypes that they were seeing, and not so much AKT, for example. So here, what I'm going to be talking is about, again, AXL-mediated bypass resistance to EGFR-targeted therapies. Again, if we target EGFR, what is AXL inducing downstream to enable survival and further malignant phenotypes? And to do that, what we have paired cell signaling or phosphoproteomics data with phenotypic measurements across matching conditions, in which we're using PC9 cells across different conditions in which AXL signaling is perturbed. to kind of map what are the signaling components driving those phenotypes. So in the way we have perturbed AXL downstream signaling is by, again, we block EGFR, and then we have generated a panel of PC9 AXL mutants cells, in which for each cell line, we have mutated an intracellular tyrosine of AXL into phenylalanine. And the reason we've done that is because phenylalanine can be phosphorylated. It's a mimetic of tyrosine, but like that lacks the side chain that can be phosphorylated. And hopefully what this will induce is specific downstream perturbations that affecting AXL signaling that will have phenotypic consequences. So again, we inhibit EGFR through erlotinib, and then we hit all of these cell lines with AF154, which is an AXL-activating antibody, and induce this perturbed AXL signaling. And the reason why we're using this AF154 antibody is because this way we make sure that the is very, very specific to AXL, and this way we make sure that AXL is actually active. So we've collected a bunch of phenotypic data with these cell lines. We're using InQSight. We tracked cell viability and cell death over 72 hours. And here you can see how the cell viability increases when we activate AXL. And even though it's not very drastic, the changes that we're seeing, what makes it relevant or like encouraging is that we see different effects across different point mutants. For example, A21 seems to have an important effect, since it looks closer to kinase death and knockout than 698, for example, and 698 would look something closer to wild type, indicating that this phosphosite may not be as important. And the same interpretation is valid for the cell death data here. Okay. Another phenotype that we've studied is migration. Someone in the lab observed that when these are just PC9 cells growing in culture without any treatment, but when we treat them with erlotinib, they form these clusters, these cell clusters, meaning that they need this cell-cell contact to survive, or at least try to survive in the presence of the drug. However, when we hit the cells with the AXL-activating antibody after treating them with erlotinib, this abolishes this effect, and we see how they're spreading out again. This is just a PCA analysis showing the different phenotypes that we have. And as you can see, we have 4 phenotypes in which 2 oppose to migration oppose. So this migration refers to a cell wound scratch assay in which like we perform a wound in the middle of the well, and then we still track their migration inside the space that is left by the wound. And this opposes the cell life island effect, and then viability opposes apoptosis, and this is clearly pictured in this loadings plot. And then in the scores plot, we see how the PC1 represents the fitness axis, in which here we can see clearly the untreated cells clustering together, positively weighted across PC1. All the way to the left, we see the erlotinib only treated cells, and we see how when we treat the cells with the AXL-activating antibody, they shift from left to right. So from unfit to fit, except from knockout here, which hitting the antibody even looks like that it's getting worse, and kinase death, consistent with an AXL specific effect. And then, so again, what we want to do is, given this cell signaling state of these PC9 cells or mutants, can we predict these phenotypes that I just described? And to do that, we could just use standard multivariate regression models such as PLSR or Lasso. However, what we've been working on in the lab is in developing this computational method or clustering method to cluster peptides before performing the regression. And how this works is by clustering peptides based on their phosphorylation signal, so the data, but also their peptide sequences. And once the clusters are formed, then we can use the cluster averages to run a regression model of interest. And the hypothesis is that in adding the sequence information, we improve the model performance. Very briefly, how this algorithm works: it's an expectation maximization algorithm that models the phosphorylation signal using a Gaussian mixture model, and the peptide sequence we provide two options, either using a binomial method or a PAM250 method. The binomial method basically takes all the sequences in each cluster, generates a frequencies matrix, and compares each of them individually, each cluster of sequences individually, to a background set of sequences that were collected from Phosphocyte Plus, and were also like mass spectrometry generated. And then we use a binomial distribution model to generate this binomial probability matrix that tells us what is the probability in each cluster of finding each amino acid across the peptide position. And then for each peptide, we get a final score, which is the probability from the data comes from the Gaussian mixture model, and the probability from the score from the sequence comes from, we look at a particular peptide, we check what particular amino acids is in that are in this peptide, and then we find what is the probability of finding each particular amino acid across all positions. We just take the average of all the probabilities found in the binomial probability matrix for each cluster. And then we multiply importantly, we multiply this to a weight, which allows us to scale or prioritize either of the two information sources. So a weight of 0 would mean that we're only using the data or the phosphorylation signal, and a very, very large weight means that we're only prioritizing the sequence. But we want to use something in between because we care about both. And then the second method is we just use a PAM250 transition matrix to calculate the distance between the current peptide that we're evaluating to every sequence in every cluster. And then we take the mean PAM250 score for each cluster, and this will be our final sequence scores. Once all of these scores are computed, when each peptide, we have the probability of being in every cluster, then we update the GMM parameters and cluster sequences using weighted cluster assignments, because this is a soft expectation maximization method, not so there's partial membership of each peptide being in each cluster. And this is how the model performs. As I was saying before, the idea is that by incorporating the sequence into the model, we will improve model performance. Here I'm showing the prediction performance of using PLSR without clustering up front. This is using k-means. Sorry, I'm not labeling. I'm not sure. I forgot the label, but this is using k-means. We see that it works for migration and viability, but it does very poorly with apoptosis and the island effect. This is with GMM, so clustering with GMM and then running PLSR, and we see that it performs rather poorly. And this is the result with our co-clustering method, where we see that it performs really well across all phenotypes. The only downside to this is that how it performs best is with 4 components, which the fact that we need to use 4 components hinders a little bit the interpretation of our model. However, it's still much more easy to interpret than just having hundreds and hundreds of peptides in our loadings plot. We would rather have these 5 clusters and see what's inside them. And these so these are the scores and the loadings, and we see how clusters 1 and 5 track really closely with viability, and seems like cluster 4 also tracks closer to migration. So taking a closer look at them, cluster 4. For cluster 4, we can take the center of this cluster, and see how it's very so the mutants Y643 and Y698 are very active. It's surprising to me that wild type is down, but this could be due to the fact that we're using the weighted average of because all of them are very low in the wild type, and then we see the effects that particular mutants have, which is something that we care about. But again, this is just an observation that is curious. But then what we saw is that many of these clusters that we're caring about, like in this case cluster 4, have many YAPTA signaling components. For example, this cluster has ABL, SRC family kinase YES1, SRC, LCK, FRK, and there's a bunch of other interesting molecules that are present in this cluster that have already been established or studied in the context of resistance to EGFR, or in the context of drug resistance in lung cancer that I'm including here. And what we can do also is with this sequence, with this cluster, we can generate a position-specific scoring matrix and display what are the residues that are enriched in this cluster. And then once we generate this PSSM matrix, we can compute the Frobenius distance or like see how similar it is to a bunch of kinase specificity assays. So to find out, okay, what kinases upstream may be regulating them. And by comparing these two sets of, so this matrix to each potential upstream kinase, we found that ABL and largely ABL in this case, because ABL really favors proline at +3, isoleucine and valid at -1, and adenine at +1, maybe regulating many of the peptides present in this cluster. And then again, cluster 4 is regulating migration. Cluster 1, so it seems to go again, be important for be high on 43, 98, and in this case also 726. It's FRK and YES1 are present in this cluster, and in our model tells us that SRC, BLK, and FRK also may be regulating them. Also present in this cluster, we see CDKs, epinephrine receptors, and other interesting molecules. And in case of cluster 5, this is where we find all AXL peptides, but we also see YES1, LIN, and FRK. Here we see that ALK and the receptor red maybe also phosphorating this a bunch of these peptides. But again, we still see a bunch of YAPTAs, signal peptides. And so as an initial validation, or like at least an initial exploration of how AXL might be modulating YAP activation in these cells, we grew PC9 wild type cells and PC9 AXL knockout, inhibiting erlotinib, inhibiting EGFR, inhibiting AXL or activating AXL. And we see how total YAP doesn't change much. This is an inhibitory phosphosite of YAP, and this is an activation phosphosite of YAP. So YAP active, YAP inactive. And we see how when AXL is present, this phosphosite is very active. When AXL is not present, it goes away completely, practically. And here it's more subtle, but we see the opposite effect, in which when AXL is not here, this inhibitory phosphorylated seems to be there. So very briefly, so by integrating a computational and experimental approach, we're able to predict AXL-mediated cancer phenotypes simultaneously in our in vitro system. We have shown that incorporating the sequence information into the model, and it enables more accurate predictions and makes it easier to interpret the clusters that we're generating by looking at potential upstream kinases that may be regulating them and therefore driving resistance. And we have predicted that, or the model predicts that the YAPTAZ pathway and their upstream kinases, ABL and SARC-related kinases, are driving cell viability and migration in PC9 cells. And our next steps are further validating our model predictions using target inhibitors for these kinases and see if actually the clusters that we think that they're regulating, when we treat them with these inhibitors, their signal goes down. We are also very interested in checking, so the YAPTAZ pathway is a very dynamic and canonical pathway that controls many things. And one of the things that it controls is cell confluency. So we're also interested in playing with cell confluency levels, or also mechanosensing by changing the extracellular matrix compositions, and see how these may be affecting, how these may be affecting resistance. And also we're very interested, or we've been working a lot already on analyzing the NIH CBTAX phosphoproteomics data in lung adenocarcinoma patients. So with that, thank you very much for listening, and I'll be happy to take any questions if there's time.